organized into chromosomes.
Okay, good.
Ribosomes are of ATS types.
Very good. Yes, most of the points you have covered it up. See the eukaryotic cells before we enter into the topic. So what you can remember is, so you can say, look into the backdrop of the cells. See eukaryotic cells, they are going to have a true nucleus. So eukaryotic cell has a true nucleus, and they would be having a true nucleus implies they have a nuclear membrane and nucleolus. Okay, so nuclear membrane is present and they also have nucleolus, nuclear membrane, or you can also call it as nuclear envelope. Okay, because of which we call it as eukaryon, true type of nucleus. Eukaryotic organisms, they have a true nucleus with nuclear membrane and nucleolus. They also have double membrane bound cell organelles like mitochondria, Golgi complex, endoplasmic reticulum. Then they also have the chloroplasts, lysosomes. They're all double membrane bound cell organelles are found in eukaryotic cells. Then eukaryotic cells, you can also notice that they have uh, the eukaryotic cells. One more feature that you notice is they are having ATS type of ribosomes with a larger subunit of 60S and a smaller subunit of 40S. And as your friend here has sent a point that they also undergo mitotic and meiotic cell division. So you notice that in case of this eukaryotes. Okay, so let us proceed with the next slide. See, eukaryotic cells, they have a well organized membrane bound nucleus and cell organelles are found. They have a well organized membrane, nucleus, membrane bound nucleus and cell organelles, you can notice. Okay, so in this slide, you can see a protist cell, amoeba. It has a well organized nucleus with a membrane envelope, and it also has cell organelles, double membrane bound cell organelles. Fungi is also a eukaryotic. See, only one kingdom is prokaryotic, kingdom Monera. Remaining four kingdoms, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. All the four of them, they are composed of eukaryotic cells. Even we are made up of eukaryotic cells from kingdom Animalia. Even human beings are made up of eukaryotic cells. So we have a nuclear membrane bound nucleus and cell organelles. So presence of membranes gives compartmentalization of cytoplasm. There is a clear compartmentalization of cytoplasm, like mitochondria is separate, chloroplast is separate. So it gives a definite boundary for different functions and different structures. Okay, so the presence of membrane gives clear compartmentalization of cytoplasm. So the eukaryotic cells, they have a complex locomotory and cytoskeletal structures. So that is also one of the features. So they, in case of bacteria, they have the pseudopodia, a type of locomotion. The false foot is formed, pseudopodia, through which they move. So amoeboid movement that you notice by the formation of pseudopodia. So that is there. Some of them might have cilia for locomotion, or they might have flagella for locomotion. So they have different complex locomotory and cytoskeletal structures. So their genetic material, they are not naked, they are organized into chromosomes. So you find chromosome structures with centromere. So their genetic material of eukaryotic cell is organized into chromosomes. That is another part that you have to remember about. So cell organelles in a typical eukaryotic cells, they consist of cell membranes, cell wall. So cell wall should be above cell membrane, endomembrane system, that is endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles. They are all considered to be the endomembrane system. Within the membrane system, endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles, they are all endomembrane system. So they also have mitochondria, plastids are there, where they have this coloring pigments. So ribosomes are there, cytoskeleton, cilia and flagella, centrosome and centrioles in animal cell. Okay, nucleus they have, and microbodies. So these are the various components of a animal cell, an eukaryotic animal cell. Now the various components of a eukaryotic plant cells are, so they have this cell membrane, cell wall, endomembrane system, mitochondria, plastids, ribosomes, cytoskeleton. Centrosomes and centrioles are absent in case of a plant cell, but in animal cell, here you can notice it in an animal cell, and they have this small finger-like protrusions on their cell wall, which we call it as microvilli or brush border. Okay. So cell membranes. So before that, we'll study the type of plant cell. This is the plant cell that you can notice. So they might give you in examination, draw a neat label diagram of ultrastructure of plant cell. So you have to draw this diagram of it. So what you can notice here is a prominent vacuole is there, a large vacuole is there. So, and the outermost membrane of this vacuole, we call it as tonoplast. So the outermost membrane of the vacuole, we call it as tonoplast. So this is the vacuole, so which is having its membrane tonoplast, and nucleus is there with a definite nuclear membrane and nucleolus. Nucleoplasm, the fluid part of nucleus, we call it as nucleoplasm. The nuclear membrane, we also call it as nuclear envelope, and you can find in between them minute openings. So this minute openings are called as nuclear pore. Okay, then you also find the endoplasmic reticulum, and you also notice the Golgi apparatus. Middle lamella is found within this cell wall. You notice this middle lamella. Cell membrane is there. Then they have mitochondria. Chloroplasts are present. So peroxisome, cytoplasm, microtubules. Then they have the cytoplasmic connections through plasmodesmatal connections. So you find both smooth endoplasmic reticulum and rough endoplasmic reticulum. Lysosomes are also noticed. In case of a uh, plant cell, ultrastructure of plant cell, they have a cell wall, so which is also having a middle lamella layer. Then cell membrane is there. So they are having a large vacuole, okay, which is covered by a membrane called as tonoplast. They have a true nucleus with a nuclear envelope and nucleolus. So within the nucleoplasm. So the nuclear envelope has numerous minute opening called as nuclear pores. Then you also find rough endoplasmic reticulum and smooth endoplasmic reticulum. Golgi apparatus is noticed in this plant cell along with chloroplasts. Chloroplasts are present for photosynthesis, they are required. Peroxisomes, lysosomes, cytoplasm, mitochondria, ribosomes, they are all noticed in case of a plant cell. So plant cell, the reserve food material is starch. In the animal cell, the reserve food material is glycogen. So plant cells do not have centrioles. Because they don't have centrioles, during cell division, there is no formation of aster. Okay, only in animal cells, if animal cell division, you find the formation of asters. That is because they have centrosomes. So centrosomes are absent in case of the plant cell. This is a ultrastructure of a plant cell. Next, if you notice here, it is the ultrastructure of an animal cell. So you can notice that vacuole is very small or reduced, whereas in a plant cell, the vacuole was very large. Okay, and you do not find chloroplast in a animal cell. So cell wall is absent. Important feature is they don't have a cell wall. They directly have a cell membrane. Okay, cell membrane is present, and you find on the cell membrane numerous finger-like protrusions, which we call it as microvilli or brush border. You already know that microvilli or brush border, they are help in increasing the surface area available for absorption. Okay, so they also have the centrioles. Centrioles are a reason for the formation of asters during cell division. So they don't have chloroplast, but they have centrioles. They have a true nucleus with a nuclear envelope and nucleolus. Within the nucleus, you find this fluid, which we call it as nucleoplasm. So nuclear envelope has numerous opening called as nuclear pore. Okay, and they also have mitochondria, Golgi complex, lysosomes, endoplasmic reticulum. Okay, so they have rough endoplasmic reticulum as well as smooth endoplasmic reticulum. Also, you can notice ribosomes. So the reserve food material in case of animal cell is glycogen. So that is another important feature that you have to remember about. So again, as I told you, they might ask you to draw a neat label diagram of ultrastructure of animal cell. So you should be in a position to write. So the assignment that you have for this topic is so to differentiate between plant cell and animal cell. So you have to write at least 7 to 8 differences between plant cell and animal cell. Okay, so that is your assignment, which I expect you to submit by 1 or 2 days because the topic is quite small topic. So do it by Wednesday. You can submit the topic to me. Okay, so you have this assignment of the differences between plant cell and animal cell. I've already listed out a large number of differences, so you have to list it out. Okay, then the next thing that we have for study is the cell organelles in eukaryotic cells. The first thing that we are going to study about is cell membrane or plasma membrane. Okay, so the chemical studies have been done on the human RBC, that is red blood cells or erythrocytes. So they show that cell membrane of RBC when they have studied, so they notice that it is composed of two layers of lipid or lipid bilayer, proteins and carbohydrates. So when they studied the cell membrane composition in case of human RBC, they noticed that two layers of lipid is there, and they also noticed protein and carbohydrates. So the lipids mainly what they noticed was phosphoglycerides, which have outer polar head and inner hydrophobic tails. So the non-polar tail or saturated hydrocarbons is protected from aqueous environment. So the lipids, they noticed it to be phospholipids, phosphoglycerides. So they have an outer polar head and inner hydrophobic tails. So the outer polar head is hydrophilic. So the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. That is what they noticed. See here, this is what is the fluid mosaic model of plasma membrane, which was put forth by Singer and Nicholson. So we'll be discussing about that. So in the human RBC, the membrane, the plasma membrane that they studied, this is what they noticed. So the ratio of protein and lipid varies in different cells. In human RBC, the membrane has 52% protein and 40% lipids are there. So human RBC, they have noticed that the membrane has 52% protein and 40% lipids are there. So based on the whether they are easy to extract or not, the membrane proteins are of two types: integral proteins, peripheral proteins. So integral proteins are partially or totally buried. See now this type of proteins, they are partially or totally buried in the membrane. Peripheral protein, they lie on the surface. See this pink colored one is the peripheral protein. They are easy to extract. Whereas the integral protein, which is partially or totally buried in membrane, they are difficult to extract. And you might notice this, the lipid, which is phosphate head they have, which is polar or hydrophilic. So they have a phosphate head, and they have this fatty acid tail, okay, which is non-polar, and they are hydrophobic. So you notice this phosphobilipid layer, which has a phosphate head, which is polar phosphate head, and they are hydrophilic, whereas the non-polar part, which is hydrophobic, they are made up of fatty acid tails. So you notice this two layers of phospholipid layer. So biphospholipid layer, proteins are here. Some of the proteins are peripheral and they are easy to extract because they lie on the surface of the membrane. Some of the proteins are partially buried in the membrane or completely buried in the membrane. So they are difficult to extract. So these are the integral proteins. So based on the ease of extraction of the proteins, they are of two types: integral proteins and peripheral proteins. Peripheral proteins lie on the surface of membrane and they are easy to extract. Okay. In human RBC, what you notice is the membrane is having 52% protein and they are having 40% lipids. So that is what you notice in case of human RBC. So the cell membrane, to explain the cell membrane, the fluid mosaic model of cell membrane was put forth by two scientists, Singer and Nicholson. So they have put forth this fluid mosaic model of cell membrane. According to their model, the quasi-fluid, that is, it is semi-fluid in nature of lipid. It enables the lateral movement of proteins within the overall bilipid layer. So this ability to move within the membrane is measured as its fluidity. So when we are talking about this fluid mosaic model, so according to this, the quasi-fluid nature of lipid, the lipid, which is quasi-fluid or semi-fluid nature of lipid, makes it possible for lateral movement of proteins within the overall bilayer. Okay, this ability of the proteins to move within the membrane is measured as its fluidity. So it is not rigid, it is fluid, and proteins can laterally move within this lipid layer, overall lipid layer. So this ability to move within the membrane is measured as its fluidity. The functions of the cell membrane are they help in transport of the molecules. The membrane is selectively permeable to some molecules present on either side of it. Okay, so the transport of the molecules they help in. The membrane is selectively permeable to some molecules present on either side of it. So now you understand how the cell membrane or plasma membrane is selectively permeable and it is semi-permeable. I had already discussed about in the functions of plasma membrane when we discuss the prokaryotic cell. So the transport of the molecules are helped by this cell membranes. So the membrane is selectively permeable to some molecules present on either side of it, that is extracellular space, intracellular space. Either side of extracellular space is outside the cell, intracellular space is within the cytoplasm. Okay, within the cell. So there is a movement of molecules present on either side of extracellular and intracellular space, selectively permeable to such molecules are there. Okay, then you also notice due to the fluid nature, the plasma membrane can help in the cell growth. If it was rigid, it might not have helped the plasma membrane. So due to the fluid nature of the plasma membrane, so the help in cell growth, formation of intercellular junctions. There are different types of junctions that you might have noticed: the gap junction, the adhering junction. So we have discussed about that, isn't it? So formation of intercellular junctions, secretion, endocytosis, cell division. So all these are possible because of the fluid nature of plasma membrane. They not only help in cell growth, they also help in the formation of intercellular junctions. They also help in secretion. They also help in endocytosis, and they also help in cell division. Okay. So these are the major functions of the cell membranes. So what you have to remember about the cell membrane is that so cell membranes, the ratio of protein and lipid varies in different cells. In human RBC, the membrane has 52% protein and 40% lipid. So as per this cell membrane that they have studied in RBCs, they have noticed that there is a two layer of lipid layer. So phospholipid layer it is. They are having a circular spherical head, which is the polar hydrophilic phosphate head, and they have a non-polar hydrophobic lipid end, that is the fatty acid tail. So they have phospholipid layer. So within them, you find two types of proteins based on their extraction. So we call them as integral proteins and peripheral proteins. So peripheral proteins are easier to extract when compared to integral proteins, which are partially or completely embedded in the buried in the plasma membrane. Okay. Okay, so the fluid mosaic model of cell membrane was put forth by two scientists, Singer and Nicholson. According to that, they said that the cell membrane is made up of phosphobilipid layer, and they have a polar end which is hydrophilic, and they are made up of this phosphate head, and the non-polar end which is made up of lipids, they are hydrophobic in nature. And within this phosphobilipid layer, you find large number of proteins are there. So some of them are peripheral proteins and some of them are integral membrane proteins. So there is a lateral movement of proteins within this uh, f- phosphobilipid layer. So that gives it a quasi-fluid nature of lipid is noticed, which enables the lateral movement of proteins within the overall phospholipid bilayer. This ability of the proteins to move within the membrane is measured as its fluidity. And we had discussed about the functions of cell membrane. They help in transport of the molecules. The cell membranes majorly help in the transport of the molecules. The membrane is selectively permeable to some molecules present on either side of it. When I'm talking about either side of it, it implies extracellular space and intracellular space. Extracellular space is outside the cell, intracellular space is within the cell, that is within the cytoplasm. So it facilitates the movement of molecules both outside and inside. Okay, and they are semi-permeable in nature. So due to the fluid nature of the plasma membrane, it can help in cell growth, formation of intercellular junctions, secretions, endocytosis, and cell division. So they are all helped by this fluid mosaic model of this plasma membrane. Okay, so in the next class, we'll be discussing about the types of transport and other cell organelles. Fine. So do you have any doubts, dear students? Any doubts or clarification?